Chapter 23 takes us to our discussion of Mendelian genetics, or the study of how characters are passed on from parents to offspring. Here are our learning objectives, starting with the first four. Well, I'll start with the first one, which is summarizing Mendel's four hypotheses. Well, this begs a couple of different questions. One is, who was Mendel? Gregor Mendel was an Austrian monk who became a father decades after his death. And that probably has you scratching your head, wondering what I could possibly mean by that. So who was Gregor Mendel? As I said, he was an Austrian monk. He lived in the 19th century. He was born in Brno, Moravia, to an Austrian family, German-speaking. Brno is now in the Czech Republic. His parents were farmers, but they did not have very much money, and he was a very bright young man, and he wanted to study. And the best way for someone in the 19th century to do that was to join a monastery. So he became an Augustinian monk, and being interested in science and having experience in farming, he was assigned to work in the gardens of the monastery, where he planted lots and lots of garden peas. Garden peas became an experimental model organism for him, and he made a lot of discoveries that went more or less unnoticed in the field of genetics. So he is considered to be the father of genetics. So that's how he became a father decades after he had died. It wasn't until around 1900 that his discoveries from the 1860s were rediscovered and their importance truly acknowledged. And you're probably wondering which of these guys is Gregor Mendel, so I'll let you know that that's him right there. He was also on the first slide. Okay, so now that we know who Mendel is and what he did a little bit, let's get into the details of how he did that. He was curious about how traits were passed from parents to offspring, and he was interested in looking at testing a certain hypothesis. There were two competing hypotheses. One was what was called the blending hypothesis, which is the idea that when two parents produce offspring, that the information to produce a new child or new progeny, offspring, were somehow blended, kind of like if you mix blue and yellow paint together, it makes green. A contrasting hypothesis to that was the idea that hereditary information was particulate. It wasn't just a matter of blue and green, blue and yellow being mixed to make green, that there were actually discrete particles that would be passed on from parent to offspring. So what Mendel did is he demonstrated how the particulate model was better supported than the blending hypothesis. And he was very meticulous, made very careful records. He used the scientific method very well. So that is how, well, for one, he was able to show these things so clearly, and two, how even though he had died many years before the value of his discoveries was acknowledged, they could still be acknowledged because he left behind these documents for future generations. So what he did was he was looking at certain features that were variable among individuals, and he called these features characters. And within a certain character, there were certain character states or traits, the most familiar of which that we will talk about first are flower color. So purple and white were two different traits for the character of flower color. And it wasn't just flower color that he looked at, but we're going to talk about that first. So Mendel could obtain a lot of different types of varieties of peas, but they were also, pea plants were a good model organism for a number of other reasons. He could grow them up fairly quickly in a matter of weeks. He could produce lots and lots of new peas and get seeds, flowers, plants, everything that he needed. He could produce in a fairly short amount of time. Pea plants also don't take up a lot of space, and the number of offspring, the number of peas and pea pods and seeds, was a very large number. It wasn't like he was trying to breed elephants or something that gestate for 22 months and then only give birth to one baby elephant at a time, require a lot of space. He was also able to control the mating of his pea plants. So they could either self-pollinate or he could cross-pollinate them. So pea plants have both male and female organs, structures within the flower. So you can actually cause them to fertilize themselves, or you can manipulate them so that they cannot fertilize themselves, and you can direct how what pollen goes onto which stigma to produce a new offspring, the next generation. So it was a fairly simple process, although it looks very painstaking in here, where he could take the scissors and cut the stamens, which produce the pollen, which contain the sperm, and you could brush pollen from one flower onto the stigma or the carpel of another flower and direct the mating. So in his first experiments, he took true breeding purple flowered plants and true breeding white flowered plants, and he crossed them. He put white flower pollen onto purple flower plants and purple flower pollen onto white flower plants. And what did he get? He got a bunch of peas. And he planted those peas, because those peas are seeds. And what did he get? In the first generation, offspring generation, called the first filial or son generation, S-O-N, he found that all of the offspring were purple flowered. The first generation he called the P generation, for the parental generation. So he was tracking characters that had two very obvious, distinct forms, purple and white, nothing kind of in between. He also used these varieties that, as I said, they were true breeding, meant that if you self-pollinated them, they would only produce the characteristic of the parent. So white flowered plants that self-pollinated would only yield white flowered plants, the same for the purple flowered plants. So by crossing the true breeding parents, this process is called hybridization. He obtained those offspring in the first filial, or the F1 generation. Remember, they were all purple. And then he did some self-pollinating and some cross-pollinating of those F1 hybrids to produce the F2 generation. So these are like the grand peas of the grandparent generation, the P generation. And what did he observe? When he crossed the F1s, he found that the white trait had returned. Most of the F2 plants had purple flowers, but there were some white, and this was in a ratio of about 3-to-1 purple to white flowers in the F2 generation. So he crossed purple and white true breeding parents to get all purple flowered plants in the F1. But when he self-pollinated that F1 generation, lo and behold, that white flower trait reappeared in about a quarter of the plants. and about 3/4 of them were still purple. Okay, so he reasoned that only the purple flower factor was affecting flower color in the F1 hybrids. So somehow, because that white flower color was clearly present, or it wouldn't have reappeared in the F2 generation, somehow that purple color was able to mask the white flower color. So he said the purple color was dominant and the white color was recessive. And what he saw was that the white of the F2 generation, it was just as white as the parent generation. There was no dilution, there was no destruction. It was just the same, just as the purple flower color. They were just as purple. They weren't some sort of intermediate pinkish color or something like that on the F1 or the F2 generation. So he observed this not just in the flower color, although that's the most famous example, but in a whole bunch of other characters that had two different traits. Nowadays, this heritable factor that Mendel called, we would call it a gene that produces these appearances. But Mendel didn't have that vocabulary. Mendel didn't know about chromosomes and chromatids and things like that, and mitosis and meiosis. So, Mendel's had 4 hypotheses that make up what we now call Mendel's law of segregation. So, here are the 4 hypotheses: that each individual has 2 factors for each trait, or 2 genes, 2 copies of a gene for a particular trait. When the gametes are formed, the sperm and the eggs, those factors are going to separate, and we know that now happens through meiosis, which we just talked about. So each gamete is only going to contain 1 factor from each of those pair of factors, and then when fertilization occurs, when a sperm and an egg meet, and a new individual is formed, they're going to inherit 2 factors, 1 from each parent. So this is how he accounted for the 3-to-1 ratio observed in the F2 generation. And I'll sort of demonstrate that here in a second through a diagram that we call a Punnett square. Now, Punnett square is named after a different scientist named Reginald Punnett. And in these Punnett squares, something that we're going to see again and again is that a capital letter is going to represent a dominant allele or form of a gene, and a lowercase letter represents the recessive allele or recessive form of a gene. So here we can see that in the P generation, the purple flowers had 2 copies of each of that flower color gene. So did the white parents. When they produced their gametes, they could only contribute either a big P or a little p. But because they were true breeding, they only had 1 form of the gene. They had only 1 form of the gene to pass on, either purple or white. So in that F1 generation, all of the progeny were purple because the purple color masked the recessive white allele, white form of that character, the white trait. But they still had it. So then what happens when we self-fertilize these F1 plants? Well, all of them are going to either give a purple allele or a white allele to the progeny. And 3 of these combinations are going to give us a purple flowered plant, and only 1/4 of these combinations, or 1/4, is going to give us a white flowered plant. So this explains this 3-to-1 ratio. And this thing down here is what we call the Punnett square. So the way it works is we put in the potential genotypes, the potential genetic makeup of the gametes onto the squares on top of the box. So each flower could either give a purple allele or a white allele to the progeny. So here are the potential gametes from the sperm, potential gametes from the eggs, and when we show what the offspring should look like, this one's going to have a big P and a big P, and it's going to look like a purple flowered plant. This one's going to have a big P and a little p, and it's still going to look purple, though it has that small p allele, but it's masked. Same thing for this one down here, and then it's only in 1/4 where they get both little p characteristics from the parents, and they're going to appear white. Okay, so here's Sir Reginald Punnett. I think he was knighted. Let's do a Punnett square with his wife, Evelyn Maud Froude. That's not actually her, but I couldn't find a picture of her. They were married, and if they had children, we could make some predictions about them. Now, another gene that's commonly used in a human example is the gene for eye color. Brown eyes are dominant to blue eyes. So brown-eyed gene will mask the appearance of the blue-eyed gene. So if you have blue eyes, it's because you've inherited that gene from both of your parents. If you have brown eyes, you either inherited it from both of your parents or from just one of your parents, and you could be masking a blue-eyed allele. But so it looks like Reginald has brown eyes, but if he was had a blue-eyed allele that was masked, and his wife had blue eyes, the likelihood that their children would have brown eyes is 50%, or 2 out of 4. And the likelihood that their children would have blue eyes is also 50%, or 2 out of 4 times. So this is how a Punnett square works. It helps us to predict the chances of the offspring having a particular appearance. So I don't know if we're quite at a point where we can start making mental Punnett squares when we see attractive potential mates, but hopefully by the end of this chapter we'll be able to do this.